Welcome to AP Biology. Today's presentation is going to be on the control of the cell cycle. We have several cells in our body that reproduce at faster rates than others. As an embryo, we have a super fast rate of reproduction, primarily because we don't have to grow in between, so you can almost eliminate the G1 phase. And cells can divide about every 8 hours. When you get to an adult or an actual human that's, a child or an adult, then the fastest cells are dividing about every 24 hours. So our skin cells and our blood cells and the cells lining our digestive tract. So we have some different types of cells that divide at a real rapid rate. And then we have cells that divide at a less rapid rate. They might divide every 6 months or every year or 2 years. And then we have cells that divide only when necessary. So when there's damage, they will come out of G0 and repair the damage and then go back into G0. And the final set of cells we have pretty much stay in G0 once you reach your adult form. So our nerve cells and our muscle cells really don't divide once they've reached their maximum size. There are two in the cell cycle process. There are two kind of irreversible points. One is when we replicate our genetic material, and the other one is when those sister chromatids separate, and those are both checkpoints, which we'll talk about in a minute. So the process of replication and the process of separating, those are kind of irreversible points of this process. All right, so there's 3 checkpoints we're going to talk about. One is called the G1 checkpoint, and it's at the end of G1. One is called the G2 checkpoint at the end of G2. And the other one's called the M checkpoint, and that takes place right before the sister chromatids separate during mitosis. All right, so the G1 checkpoint. This is the primary decision point, okay? So at this point, a signal has come to the cell and told the cell it's time to divide, and the cell has to decide whether or not it's in condition to divide. Okay, so is the cell big enough? Has it grown enough since the last time? What are the nutritional factors going on inside the cell? And what is the condition of the DNA? If all those things are ready, then we can do cell division. And if they're not, then a couple other things might happen. Either cell is going to repair its DNA if that's damaged, or it could grow some more before it actually starts to divide, or it could do what's called apoptosis, where it actually self-destructs because its DNA damage is so bad that it doesn't feel like the reproduction of that cell would be a worthy thing to do. Some of our cells are going to stay in this G0 phase. They won't leave G1. So even if they get signals to divide, they're not going to divide. So nerve cells and muscle cells, these are really highly specialized cells, and they arrest in G0. So they get to G0 and then they won't come out of G0 for the rest of your life. That's why nerve damage is permanent. If you get significant muscle damage, it's permanent. When people have a heart attack and it damages part of their heart muscle, that reduces the ability of their heart to pump blood for life. When you have a stroke and you damage part of your brain tissue, that tissue is damaged for life. It doesn't recover. Now, maybe in the future we'll be able to fix that with stem cell technology, but as of right now, we're not quite there. So This G1S and G2 is all part of interphase, which is 95 to 99% of a cell's cycle, or even more than 99 in many cases. But this is when a cell is doing its job, okay, interphase. Most of this interphase is G1. The way it's divided up right here, G1S and G2, that would really only look like that in fast dividing cells. So in a really fast dividing cell where we have division going on every 24 hours, you would have a G1 that would take about 8 to 10 hours, S that would take about 8 hours or so, and then G2 actually would be quite a bit less and only be a few hours before the cell divides. So, well, let's go back. In G1, basically we have, and the G doesn't stand for growth, but a lot of growth goes on during G1 because you just cut yourself in half, okay? And so in G1, or the gap, the first gap, okay, the cell is basically going to be doing its job and growing, okay? This is where cells will go into G0. And then in the S phase, if we get the appropriate signals that it's time to divide, that's where we will replicate our DNA, if the DNA is intact and ready to go. And then G2, basically what you're doing is you're basically just getting ready for cell division, producing some organelles that only have individual, like the Golgi or centrioles. We have to duplicate those before we can move into cell division. And basically just getting ready for mitosis. Okay. Part of the S phase is replication of DNA. Replication of DNA is super important, and it has to be done correctly. In the S phase, we take apart the original copy and we make 2 new copies so that each daughter cell can get a complete copy. And there are a lot of mistakes that have to be corrected. And so in the end of the S phase and during the G2 phase, you have a bunch of proofreader enzymes that are going to go along and correct mistakes so that we get the error rate down to a really low rate. About 1 in 1,000 or about 1 in 10,000 are screwed up initially, but we do get that rate down to about 1 in 100 million bases after we do all the proofreading. So there are still 30 mistakes each time cells divide, but hopefully those aren't in important places. Sometimes they are, and that's what can lead to cancer and things like that. But most of the time, the mistakes are unimportant, and so the cells continue to go on just fine. And all of this is going to lead to mitosis. And mitosis is going to be the dividing of the DNA between 2 daughter nuclei, which we'll talk about in the next video.